you for joining us in this special commemoration. Now, as a mark of our deep respect and gratitude, I now invite our Honorable Director Madam, Dr. Bhanu, and Research Coordinator, Dr. Hegde, and Academic Coordinator, Dr. Sridhar, to kindly come forward and offer a floral tribute by garlanding the portrait of Dr. Lalji Singh. Everybody, please stand up. Thank you. Now to formally open today's event, I now have the pleasure of inviting our respected Director Madam to deliver the welcome remarks and set the tone for this important lecture.
Thank you, Dr. Sonu Gandhi. A very good afternoon to everyone. Greetings of this afternoon. Many thanks to the speaker, Dr. Bhanu Telugu. I, on my behalf and on behalf of NIB fraternity, welcome you officially at this important occasion when we are celebrating the science life of the former and the first chairman of Scientific Advisory Council of this institution, Dr. Lalji Singh. 5th July happens to be his birthday. So this institute celebrates his life and his contributions. And as Dr. Sonu Gandhi has elaborated what all he has done for the nation and across for the science, across the globe for the science. So in this series, we look forward for, renowned scientists who have made dent into their own areas. And that's how we keep talking to people, reach out to people, so that, as rightly was said by my colleague, that it is actually not only to motivate youngers, of course, that's a prime thing. We also wish to stay motivated and kind of keep the legacy on. With that, somebody would be there to officially tell about, Dr. Bhanu. But good old friend, Dr. Bhanu, a lot of which converges at one point, which makes me feel very good to know Dr. Bhanu. Besides his work, and we together in an area of reproductive biology. Not only that, he has hosted me to his lab at Maryland. Very impressive thing, colleagues, when he has shown me the transgenic pig which he made, and especially I could really understood how these transgenic facilities should be maintained following on the rules and the norms. That was very good. Me and one of my doctoral students, when we student with there. So maybe it's a coincidence, or fortunately, one of my PhD student, when he got a Fulbright Nehru fellowship, so he hosted him in his lab for a year and a long, and the story got it amplified in a professional connectivity. He has been our institute when I floated his name. Some of the colleagues and some of the staff who have been associated with the NIB since inception. So this name is known to a good number of people. It was Professor Redana's time when you were visiting an IAB at Mayapur. So with that, we looked, and he was also very kind to join us when Dr. Nirmalya Ganguly arranged a workshop on transgenesis. So it was not that comfortable slot for him, but still he was there to give a talk and to interact with our younger colleagues and with all of us. With I really am happy that you are here, and welcome you, and welcome all my colleagues and students, fellows, and looking forward for the talk. Thank you so much.
Thank you, madam. It is now my privilege to introduce the distinguished speaker for today's memorial lecture, Dr. Bhanu P. Telugu, a renowned scientist, educator, and entrepreneur, currently serving as Associate Professor in the Division of Animal Sciences at the University of Missouri, USA, and as the President and Chief Officer of Renovate Biosciences, a biotechnology startup based in Maryland. His research focuses on the use of advanced genome editing tools like CRISPR to develop disease-resistant livestock and custom large animal models of human diseases, bridging the gap between agricultural sustainability and biomedical innovation. Dr. Telugu was recently honored with the CRF Randy Wheelock Research Award by the Choriodermia Research Foundation for his work on developing a novel porcine model for this rare genetic eye disease. We are truly honored to have Dr. Bhanu with us today to deliver the Dr. Lalji Singh Memorial Lecture. Please join Dr. Bhanu on the stage. Thank you.
Good afternoon. Can you hear me okay without that? Okay. It's a great honor and privilege to be invited to give Dr. Lalji Singh's memorial lecture. Glad to see all of you here. As Dr. Sharma has introduced, I'm an associate professor in the Division of Animal Sciences at University of Missouri. I had to declare a conflict here, so I'm also a founder and chief scientific officer at Renovate. We use custom models, genome editing tools to make models of disease. Okay. So in our laboratory, our main focus is genetic engineering. We use the genome editing tools to use in agriculture and medicine. For agriculture, We're addressing important priorities in both animal health and food security. So I'm listing the species with which we are working here. On the biomedical side, we use genetic engineering to address critical priorities in disease, regenerative medicine, and also work on topics that are intersectional to both animal and human health, which we call as One Health. So in today's talk, I'd like to start off by introducing the topic, genome editors, how we're using them, what are the advantages and disadvantages of these tools. I'm going to kind of highlight some case studies of how we're using these for agricultural applications. And in final few slides, I'm going to talk about how we're leveraging these for biomedical applications. Okay. Our laboratory was one of the first to adapt the CRISPR-Cas editing tools in large animal species. What we have been doing is leveraging these tools to do modifications directly in the embryos. So in this case, you would harvest the zygotes and deliver the CRISPR reagents directly into the cytoplasm. In doing so, we can do precise modifications that leads to either generation of knockout animals, or you can make site-specific modifications. So this would be altering the alleles, creating new variants of these genes. And the advantages of genome zygotic editing is, as you can understand, the efficiencies are quite high. So hardly there are CRISPR tools that you deliver directly into the zygotes that don't work, right? So high efficiency of editing. When you're using these embryos for embryo transfers, the pregnancy rates are high. And another advantage, like I said, you're developing both sexes, males and females. There's no bias unless you introduce them. And this can be used to generate a herd of nucleus herd, like generate a pool of founders that you can breed and maintain the line. So very easy to make, very prolific and proficient, and very easy to make a cohort of animals. The disadvantage is, it really is like a elephant and 5 Chinese monk. So each of them finding different things. That's the tail. One thing that is the fountain and something else, right? One thing that's the fan. The problem, what we have seen is when you're delivering the reagents, it really depends on where you're sampling them. You sample the tail, you think it's a biallelic modification. You sample the ear, you find another modification, right? You sample the blood, you found a new mutation. So what we have seen by delivering the tools directly into the zygotes, we're dealing with huge mosaicism issues. Because once they start cutting, you have different alleles being propagated within the animals, right? So G0 animals, these would be the founders, are invariably mosaic. So meaning that you can't take these animals and do a test directly, so you have to maintain them, breed them over multiple generations. So this adds considerable time to the projects. So I know most of you work with mice in the auditorium here. Some of you may be working with goats, and you're working with cattle. So you can see how this can be really a problem when you're working with cattle, right? I mean, generation interval, think about it. 10 months of gestation, right? And it takes another year for them to be sexually mature. So you can't do a PhD working in cattle doing zygotic editing, forget it, right? So this is a problem I'm talking about. Mosaicism is a concern, and you have to make a cohort of animals. In mice, you can achieve that. We don't care because you can breed every 2 months and we can make a whole bunch of generations within no time. But even working with animals like pigs, oh, sorry, just give me a second. It's about 3 years. And the nearest one I would say is goats, which I know you have on the site here. It takes about 3 years to breed them across 2 generations to get homozygous of anything, right? And in cattle, it's more than 5 years. So it's a problem. So to counter that, in our laboratory, we predominantly use what we call a somatic cell nuclear transfer or cloning, okay? So in this case, what we do is you do cellular engineering. So you start with cells. This is typically fibroblast cells. This is fetal fibroblast cells. You introduce the intended genetic modification. Let's just say we want to make a GFP pig. Don't ask me why. Let's say we want to make it, okay? So you introduce the transgene. You can select for those cells, okay? Then you start with oocytes. So this can be slaughterhouse oocytes. because what we're doing is we're taking out the nucleus from it, right? We're taking the polar body, you're taking the metaphase 2 template. So you have an enucleated oocyte. You take that cell, put it into this enucleated oocyte, you fuse, you activate, and then you do embryo transfer. So in a nutshell, what happens is in one generation, you have a cohort of animals that have, all of them have one pre-screened genetic modification. There are no surprises, right? All of them have the pre-intended genetic modification, and all of them are clonally similar. So they're genetically identical. So you have a population of animals that are genetically identical, like your infrared mice, that have the same genetic modification. So you can readily go into your experiments.
The same method that you use for dolly.
Yes. Right? It's the same method that we use for dolly. So if everything goes well, you end up with a GFP pig, right? Again, no surprises here. And the main advantage with SCNT is the timelines are more manageable. Instead of 3 years for pigs, now you're talking about 6 to 7 months. So about 2 to 3 months for genetic modification, and pregnancy, we can't alter it, 4 months of pregnancy, right? So about 7 months. So you can have enough animals that you can do a study. When you think about it, this is actually a faster timeline than if you have to work with mouse models. Remember, 3 generations of breeding to get your homozygous animals, right? In 7 months, I can make you a cohort of pigs. So what we have done is, with the pigs, at least with the SCNT approach, for the very first time, we can actually make animals much faster than the mouse models, right? So the limitation of not having these animals is not, it's no longer a limitation anymore. So if you have a question that requires a large animal model, right? I think we should go for large animal model. And there are several examples of why we want to have a pig model for a particular disease, right? So advantages, all offspring have the intended genetic modification. All of them are clonally identical. And because they're genetically identical, it's almost like you're working with inbred animals, right? The disadvantages, as you can understand, efficiencies are not great. So our laboratory, we routinely on every week, we do like 2 rounds of cloning, right? So our efficiencies are somewhere between 5 to 10%, meaning that if you put 100 embryos, we get 5 offspring, which is actually really good, right? But then a lot of pregnancy failures also, right? And not to mention, there are reprogramming defects, because what you're trying to do is you're trying to reprogram the entire epigenome as well, right? Because of the cloning process. So you're talking with fibroblasts, you need to take them back into the developmental clock and start the development all over again, right? So in doing so, we see some of the animals, they have epigenetic defects. So with that said, this is fairly a routine process. If you want to screen a mutation, you want to study a disease, this is probably the best way to go forward, okay? So speaking of applications, now that we have the basics set up, how can we leverage this for agriculture and biomedical applications? So from an agriculture standpoint, I feel like these genome editors, we call them precision agriculture, right? These are basically a natural evolution of all the breeding technologies, right? So remember, whenever you're breeding, you're intending to create a genetic modification, right? The act of mating 2 individuals, that's the genetic modification itself. So you're genetically modifying the animals. But what we're doing with precision agriculture is, instead of waiting for a genetic lottery to happen and breed a lot of animals and find one animal, you can actually predetermine what you want, right? So you can introduce the traits directly. So I feel like, again, we started with mass selection. We said that bull has the best traits, that cow has the best traits, so let's breed them, right? But as we are going along, we have pedigree selection. Then we have the SNPs, so we have marker-assisted selection. Then transgenics came along. And this is truly a revolutionary technology because for the very first time, you can introduce the traits that do not exist in the population, right? Then we have with sequencing, we have genome-wide selection. And I feel like genome editors are next in the ladder of precision agriculture tools. So how can these be advantageous compared to what we call a selective breeding, right? So when you're doing selective breeding, the main disadvantage, I feel, is that you are relying on traits that are already existing in the population. If the trait doesn't exist, it does not emerge in the population, right? But there is a rich diversity of traits that exist in other animals, which we cannot bring into our population. And so a very good example is, this animal right here, warthog, is a natural reservoir for African swine fever virus. You heard about this. So African swine fever epidemics, right? They come and they just wipe out the entire population. They just die like flies. But this animal tolerates them pretty well. So if we can identify what's about the warthog that causes the disease resistance, right? We may be able to bring that into our population without having to breed them, which is against species barriers, right? The same thing goes. You have Bos indicus animals and Bos taurus animals. You may have a better heat tolerance trait in our indicus animals. You don't want to be mating that to a taurine animals and kind of dilute the genetics. What if we can move the trait directly into these animals, right? So that's the advantage with these genome editors. So second thing is, whenever we're breeding, we kind of hit this bottleneck, right? So we call it a hitchhiker effect. So as long as we're selecting for one economic trait, we tend to lose other beneficial traits in the population because you're dependent upon this haplotype conversion, right? So linkage disequilibrium. So a case in point here, you look at the milk production in the United States, over a period of like 50 years, there's a 100% increase in the amount of milk that's produced by the animals. But at the same time, you can see the reproductive performance of these animals started declining, right? This is because the chromosomes, they break as units and they get inherited at a time. So as long as you're selecting for one, if there is another trait that is, another variant that is disadvantageous, that's also kind of coming with it, right? So it gets bottlenecked. So for us with genome editors, you can actually move that trait into it without having to rely on all the chance genetic modifications, right? And another one, and we alluded to this earlier, selective breeding is a very slow process. I remember when we were a child here, we started this breeding, right? Bringing in genetics from the States, and we started doing the crossbreeding with the hopes of increasing the milk production. That never came along, right? So that's why it never works, because you really need like 3 to 5 generations of breeding to actually see a marginal improvement in genetic gain, which acts incredibly long periods of time. So that said, I want to highlight a couple of examples, again, going back to the discussions here, case studies as to how we can put this into practice, okay? So the first thing is, again, this is a published study. This is the first time anybody has shown that you can go directly into the zygotes in cattle and actually make pinpoint modifications, right? So what we really wanted to do is make cattle that are resistant to mad cow disease or the prion disease. And most of you are familiar with, this is a misfolded protein disease, right? So this is a healthy protein, and in prion diseases, the proteins kind of misfold, and they form tau for this and all that. And you have different names in different species, so each of them have a variant of mad cow disease. So a better illustration of what I just said about moving those traits across species, right? So this is a good example of it. So we came across this paper, and this was looking at this population in Papua New Guinea. These people practice cannibalism. There is an outbreak of disease called as Kuru, which is a prion disease. So in doing so, there are a few people that actually are resistant to the disease, while the rest of them died. So they actually went and looked at GWAS studies and identified what the mutations are. So they identified that they actually have a variant, a glycine 2 valine variant in this population that makes them resistant to the disease. They created that mutation in the mouse models, and they challenged it. They're resistant to prion disease. So the obvious question is, well, cows have prion disease. Why don't we just change that G to a V, right? That should make them resistant. Again, that's a human trait that we're bringing into cattle, right? That's only possible with precision agriculture tools like genome editors. I don't want to belabor this, but the idea is you go here, intend the genetic modification, change those two nucleotides that would result in glycine to valine modifications. So we delivered the reagents and we did target amplicon sequence. So you basically sequence at 200,000 reads in the entire embryo. So you're not missing any modifications that are happening in these embryos. And again, a lot of work went into it, and we identified the best combination of the reagents, CRISPR tools, guides, targeting reagents that gives us the best optimal combination to intend the genetic modification. We did the embryo transfers. We did 10 embryo transfers. We got 7 cows. Again, pretty high efficiency, if you ask me. All right. And one of these animals, as you can see here, with high throughput NGS sequencing, we could identify that greater than 90% of the alleles represented the intended genetic modification. So HDR was pretty good at a high efficiency. Another example I would like to bring to, and this is pretty much the origin story of our company, is the generation of these animals that are resistant to a disease called PRRS, which is porcine respiratory and reproductive syndrome. Right? So, and the latest update is this now has received FDA approval. So this is the second animal model that is been approved by FDA for human consumption. Right? The first one is alpha-gal knockout pigs. And the problem with this is it's a very expensive and economically important disease, at least in the pig industry. Causes about $6 billion in costs annually. Vaccines are ineffective. The females, they have high rates of abortion, a lot of mummified fetuses. Males, low libido, low sperm counts. So it's purely a reproductive disease. And young and growing piglets, they have high mortality and poor growth rate. So if you're a farmer, you have a farm in you for multiple generations, and this disease hits you, there's no way around it. So you have to basically kill all the animals, shut down the farm, clean it up for 6 months, start all over again. Right? So that's why the costs are so high with this disease, and it's a pretty aggressive disease. So if your vaccines are not working, it's a very bad disease, right? It's very infectious disease. What do you do? So this is where we turn to genetic engineering and see if we could find a solution for this, right? So going into the pathology of this disease, the virus gets entry and infects macrophages in the lung. Specifically, they bind to this receptor called a CD163. And when we did some mutational screens of this receptor, we identified that exon 5 codes for one of these repeats called, exon 7 codes for one of these repeats called 5. That is exactly where the virus binds. So if you take out that 5th repeat, the virus no longer binds to it. And if the virus no longer binds, it cannot infect, so it can make them resistant to the disease. And again, going at the schematic, that's the exon we want to knock out. We tested a total of 58 different guide RNAs. So we literally spent like 2 years just working on this aspect of it. Remember, we're going through FDA for approval. And the way they regulate it is every mutation is regulated as an event, right? So meaning that all your animals pretty much have to have the same modification. If they have other modifications, they're not going to count it. There's going to be another application for it, right? So our goal has to be one specific modification. So very high efficiency. And then we also need to have very low off-targeting effects, right? So again, this is a story on if you're taking some of the stuff from your lab into the industry, these are the things we have to contend with. If Prasad wants to knock out exon 7, he would just design 2 guides and cut it off, and that's the end of the story. We can breed the animals later, right? But an FDA approval, it doesn't work that way. So we have to show that they're efficient, but they have no off-targeting effect. Right? So, yes. Yes. Yeah.
What is, I mean, in this, I mean, how to work with off-target?
So it's a very good question.
This is a challenge.
Yeah. And the way to do it is, one thing is we've seen that when you're delivering the reagents, as mRNA.
When you are selecting the site, now at that time only how you are eliminating the off targets of this CRISPR test.
So we went with all of this. So we did an in vitro screen. So I'm going to show you in the next slide. So we did off target. So we went and looked at bioinformatically. So started there.
All 10 sites and then n number of sites. Then how come you just focus on that specific site?
So we did not. We went for all of them because we don't want to be pre-selecting for it. So we basically wanted the data to tell us what's happening. So we basically went ahead and did the genetic modification, and then we sequenced, did the whole genome sequencing of those.
So tip of iceberg.
Exactly. Yeah. So that's why it was the whole experiment cost us $40 million. So for FDA approval, because we had to do all of these experiments, right? So bottom line is we have to do a lot of these range finding studies where you deliver the CRISPR, look at on targeting activity, but also look for off target activity. And you have to do a lot of dose escalation. So you have to do a lot of range finding studies. So identify the optimal dose where there's enough off activity, but low off target activity. So the idea is switch more towards low activity than switch more for high off target activity. Okay. Good question though. So then, this is another way to look at it. I don't know how much you know, but the poultry industry, the pig industry, they're all vertically integrated. So there are maternal lines, there are paternal lines, right? So there are 2 breeds that they breed to get to maternal lines, and then 2 breeds to maintain for paternal lines. So you have to do genetic modification in 4 different lines, and then breed to 2 successful rounds of breeding to get that one commercial animal that's going to the farmers, right? So it's a huge undertaking. So we did this over a period of 2 years, generated more than 3,000 animals, and maintained them at the nucleus herd, bred them, looked at all the parameters. That all went into FDA package. That took us almost 10 years for us to get approval, right? So entrepreneurs in the field who wants to take this to ag, so those are the timelines you're dealing with, and that's regulatory concerns that we have to contend with. Because ultimately, the idea is if you're going to take it to the people, you have to show that it's safe. And the only way to show that it is safe is you actually go through a regulatory approval process. Somebody looking at it and says all of this process is safe for both the animals and also for human consumption, right? All right, so last thing that we have to show is that these animals that we're generating actually have the intended phenotype. This is all good. So we take these animals and we infected these animals with the PRRS virus, and none of them showed viremia. Okay. And this is, again, histologically, you can see, one on the left is the wild type, infected with the virus. You can see massive inflammation, a lot of necrosis, right? On the right is the knockout animals infected with the same amount of virus. They're perfectly fine, right? So we basically, by modifying that one group of nucleotides, made them genetically resistant animal. Okay. So, yes?
Both of them for macro as well as the homologous is required for heterologous.
No, it has to be knockout. Right. Homozygous, yeah. So this is it's a scavenger receptor. I think it interacts with iron heme molecules. No functionality problem. They have multiple repeats of this. So one basically that's part of the second equation that I didn't show. But bottom line is that you have to show that these modifications are not harmful to the animal. So you have to phenotype the animals also. So you grow these wild type animals and these animals and show the growth rates are normal.
Stress response.
I'm sure.
So if they get stressed, all in one day, problem. If it's a metabolic, we are doing well, so metabolic is fine.
Right. So remember, these animals are raised for 6 months to market. Right. Yeah.
So what about susceptibility to other bacterial infection? 163 is scavenger receptor, will not gain immunity against many bacterial infections.
has it been checked, this peaks sensitivity to other bacteria?
So we have tested them against other viruses as well. They do not seem to be like African swine fever virus. We checked it, they are not resistant to those. I don't know if we tested against bacterial diseases. I can't really say for it. Okay.
So when you go for these regulatory approvals, so what else you have to go through, such as like all the meat quality, taste, weight, all these things? Do you have to do all these?
Yes, you do. Well, the taste is apparent because if it tastes bad, then consumers won't eat it. Then So in animal agriculture, and again thinking about it, you need two licenses, right? I feel like one is the regulatory license, another one is social license, right? So we showed, responsibly that we did every test we could and showed that these animals are safe. They're safe from the virus. Meat testing panels, we did everything. But at the end of the day, people may not choose to have this GMO. That's another thing that we have to contend with. But
This thing that's came and certain mention on the.
we don't have labeling requirements in the US. You don't have labeling requirements. No, not required. They're fighting very hard against it. Yes. Can we come back to the pan later on? Yeah, I just want to move on to plenty of slides. All right. So again, summary so far. I hope I showed you that we can use these tools for one, introgressing novel traits, and two, introducing novel allelic variants, right? Okay. From a biomedical standpoint, the most obvious thing when you think about pigs would be as disease models, right? So in our laboratory, we generate a lot of these animals for cell therapy and gene therapy research. So you have a gene therapy product that you want to develop for blindness is a good example. We make that model with that intended mutation, and then you can test the dosage and safety and all those things in the large animal model before we can go to humans. Again, from cell therapy, which I'm going to allude to this later on, that's been the focus of our lab, majority of it going forward. So I'm going to switch, most of it is going to be on the pigs going forward. So the most obvious question to you guys is, why pigs, right? Why? So the question that we contend with all the time, right? So we believe that pigs, we really need a new paradigm when we're working with this, because we're working with mouse models that form the foundational rock for foundation bed for all of the research that we do. Yet 90% of the drugs are failing at various stages of clinical trial, because most of that work is being done in the mouse models, right? So we believe that now that we have this availability to make these tailored modifications in a large animal alternative, which is more physiologically, anatomically similar to humans, right? This may provide a better alternative for disease research going forward as well. At least we should consider this as a bridge animal models between the mouse and the humans, okay? The risk is going with a drug that is not ready into human population. That is a major risk to me. Another thing is, again, setting up the rationale. The pigs, again, as said, their size, anatomy, and physiology are similar. They allow for fine-tuning of surgical procedures. So any procedure that you do in humans, we do it in pigs. No difference, right? Routes of delivery. You would be surprised that some of the drugs that you're discovering in clinical trials, they're delivered a particular way because that's the only way they could deliver it in mice, right? Because something like blood collection and everything is not that routine in the mice. You go retroorbital collection and so forth, right? So again, similar routes of delivery, meaning that the pharmacokinetics would be identical as humans. You can do serial and longitudinal samples. You can go and collect blood every day, like a cc of blood, no problem, right? Which means that you can come up with better predictive biomarkers for the disease that you're studying. Again, organ size similar. So this is particularly relevant when you're working with xenotransplantation, you're working with cell therapies, and so forth. And again, longer lifespan, which allows you to study long-term